All right, good afternoon. We are now on the Thanksgiving break lectures. I think there's going to be five of these. They're going to be numbered 22.0 was what we did in class, so they're going to be numbered 22.1 through 5. I'm not sure I'll be able to get through all of them right now. They might be posted in stages, but at the very least, let's go ahead and get started because I want you to have something to watch on Wednesday for the class that we don't have in person. At least we can do the class in video. And then we'll be ready for our final in-person new lecture material class, which will be the Monday after Thanksgiving. All right, so let's talk about things, and three things that you might not think go together. Lipid nanoparticles, butter, and candles. These all are based on the chemistry of lipids, and they actually are all related to the unsaturation versus saturation point. The unsaturated points are double bonds, and those actually have a major part to play in all of these. Now, there's something I forgot to show. At the end of that, we talked about this lipid, the DPPC, dipalmitoyl phosphatidylcholine. And we're going to again talk about the choline part, which is at the bottom right here, and it's even covered up by my head. But what we talked about is we talked about how it has two palmitate tails. Those tails just go up from ester bonds, and we're going to talk about how that all works as a theme of this whole lecture series. And so what you're going to have is you're going to have this, and remember that they work for newborn lungs because they coat the lung alveoli, right? And where it meets the air is hydrophobic, so the tails point up. The palmitates are hitting the air because they are happier in the air than in the water. And they had this example that's an incredible tribute to phospholipids. We can sort of think of the palmitates as the legs, and the legs are up in the air, and the heads are underwater, in the sense that the heads are charged, and the head group has that phosphate on the bottom, and so it's able to actually be compatible with water more than air. This was shown at the top of that, and I probably will reorganize it to do a better job of that next year. But that does remind you of where we just came from. So remember, hydrophobic things are afraid of water. That's what the word means. So they will partition away from water when they get the chance. And if you have something that doesn't have any OHs on it, anything that will hydrogen bond with water at all, any O's or N's, then you're going to have something that's very hydrophobic. And in fact, what you can do with a fatty acid is you can turn that acid group into an ester. And if you do that with just two carbon chains, you get something that's incredibly hydrophobic. It's incredibly waxy. In fact, it is a wax. If you look at honeycomb, it includes wax molecules where the bees' bodies have put together the fatty acid of palmitic acid. They've esterified it with this other carbon chain that's even longer. And this just makes something that's really long and it goes all the way away from water and repels water, in fact. So waxes are excellent water repellents. It's why you wax your car and the water beads up on it if you do something like that. And at the bottom you can see the DPPC has this very waxy part of it. The palmitate is very waxy, but it's only half waxy. The other half is more is what we call the head group, because it will like to interact with water. It's hydrophilic rather than hydrophobic. So just want to remind you, you're going through this reading, and we're talking about what the nanoparticles are made of and what the mRNA inside the vaccine is also made of. And if you look at this, the mRNA is actually on the inside, and we're talking about the outside in this chapter. And so all the hydrophobic tails come together to make this sort of two-layered shell, which is really the nanoparticle. It's the part that we're talking about this time. And so as you're reading through the thing, what you want to do is you want to look for the two tails, the 2 feet of each lipid. You see the blue things right here that have this sort of ring. The ring is the blue part, which is the head group. And then they have 2 little tails that are sticking down into the hydrophobic portion of the membrane. So when you're looking at that, you'll get a figure like this. And one of the things that I will do on the test is I'll just basically ask you things that they said in the reading about these lipids. And to be able to answer the readings, you need to be able to look at the different lipids and understand which part is sticking down and which part is sticking up. So they have different categories of lipids that make the membrane. You have, like, for example, they can even have some that are just carbon chains attached to a central nitrogen. But this nitrogen is quaternary. It means it has 4 bonds to it, and therefore it's like ammonium. It's an ammonium lipid, and they call this DDAB. Now, you don't have to memorize the names, but you have to be able to see what's important chemically about this. Look at it like an organic chemist. This is one of the lipids. You can see it's got the 2 tails, and then it's got the charged head group, so you know which way it's going to orient in the particle, the way that we've shown it. And what you want to do is you want to sort of look at each of these names, and just being able to tell which abbreviation goes with which name is very helpful. So over here you have ethyl phosphatidylcholine. Again, we see that choline thing. And actually, if you look at this, this one's EPC. So let's find EPC. Here's EPC down here. And you can see the 2 tails, but these 2 tails actually have double bonds in them. So this is going to behave a little bit differently. It's also going to have a very different charged head group on the other end. But they are drawing this nicely for you because they're putting the 2 tails down where the tails go. They're putting them together, and they're putting the head group on the other side of the molecule. So I want you to see how all these work. We've just started talking about the tails. We'll talk about all the parts of this as we move through the chapter. The other thing is, like, you can look at this in DDAB, which is this molecule right here, and DOPE constitute the commercial product transfectase. Now, if you look up and down, we don't have DOPE shown here, but it's elsewhere in the reading. The other thing to do is look at the categories that they have. They have this DOTMA and DOTAP, right? But they say they have in common that they're cationic lipids. How are they cationic? Well, this one has a positive nitrogen, and this one has a positive nitrogen. They have different linkages to the tails, so this one has esters, this one has ethers. But again, if you understand your organic chemistry functional groups, you actually can see exactly, they can talk about this exact molecule in an exact chemical way. And I want you to use your organic chemistry to understand how to read, how to associate the structure with the abbreviation. Again, not memorizing the structures or the abbreviations, but simply being able to go back and forth between the reading, where they put it in a sentence, and the picture, which is what you should have in your head when you're doing the reading. So you can see that we have ionizable lipids, and you can start to think about how those are different. Those are actually different because the nitrogens are tertiary, not quaternary. That means that they can accept or reject a proton, and their charge will be different depending on the pH. This is a huge concept, and so you want to get it right here. And in fact, if you're watching this on video, one of the things I suggest is take a break from the video at times and go to the reading and go back and forth and find this part in the reading. Find where in the reading they're using these abbreviations to talk about particular lipids. If you can do that, then you can read this paper, and that's what I want to see. So if you look up and down, you can see other things where they have the big long name, but it's usually better to be able to, you can take apart the big long name if you need to, but what's the big long name refer to? D-lin DMA. And so if we look over here, here's D-lin MC3 DMA. So this is a version of that. And in fact, you see you have several D-lin DMAs. There's D-lin DMA regular, D-lin KC2 DMA, and D-lin MC3 DMA. And finally, we have the one they show us a picture of. What's the MC3 refer to is you want to sort of take that apart. You want to compare it in the reading. And one of the things is the D-lin DMA, what is extra when they have the MC3? I believe if you look up and down, you can see that there's this, the MC3 is something in the middle here. And it's going to be related to one of the structures here. So that's the way to look at this. You're kind of decoding, and you're going in between the texts where they talk about this, and they say they had further optimization of the amine head group of the KC2 version gave you the MC3 version. So that means the difference between KC2 and MC3 must be with this amine head group that's all the way over here on the left. And in fact, you want to look at that really closely, and you just want to say, okay, what could KC2 and MC3 mean as far as what these different things are referring to? And just being able to match the label to the structure actually gets you halfway there. That's a lot of what I'm going to be testing for. So this is how to read the paper, and MC3 based lipids are all going to have structures like this. So when they say how they work, they're all going to be ionizable. And the important thing is to be able to answer that question. Ionizable versus cationic is a major piece of this paper. So basically, and now I want you to go, you have to do the work of taking it apart and picking through it and figuring out how to read a paper like this. So in fact, if you look right here, if you were like, oh, what's the difference between MC3 and KC2? They have it in this table. So you, and this is actually something that I've looked up online by just looking it up. You can feel free to do that, because we can look at this right now, and here's the MC3 on the bottom. The KC2 has a different linkage, and the D-lin-DMA has an ether linkage. The KC2 has this weird sort of double ether linkage, and you see the MC3 is actually an ester. So we're going to go back there, and there's the ester. So that's the difference between the three different kinds of D-lin-DMAs that we have. It all can be figured out if you know your organic chemistry and you go back to the structure. So, and feel free to look up things like this. Right here, you have, this is the difference between the cationic lipid, and they have DOTMA there, and you see that DOTMA actually looks like D-lin-DMA. And you can make connections like this once you are able to go back and forth. And here it is again. I brought it up so that you can look back and forth. But I want you to do this in the paper. It's why I give you the paper. I want you to be able to do this with a paper as you go through. And so if you look at the paper, you can see that different lipids are actually used in the different vaccines. One of the differences between Moderna and Pfizer is not the mRNA sequence in the middle. They both have the same optimized spike, but they have different lipids on the outside. And in fact, I'm convinced that this is part of the reason why they might react differently in different people, and we need to consider this as we are fully looking through the long-term safety of these lipid nanoparticles. It's a problem that we can address. So Moderna is this lipid up here, Pfizer is this lipid over here, and you find that in the text. And then they have this zwitterionic. That's another category to look for. And remember, zwitterionic means you have two full ions close to each other. So when you step back, the ions cancel each other out. But when you're up close, you can actually see the separate positive and negative charge. This lipid is overall neutral, but when you're close to it, you'll be able to see a plus over here and a minus over there. It's all organic chemistry applied to biochemistry. So this FTT5, and if my head's blocking it, you can look that up in the paper. This is a biodegradable version of the lipid above it. So why is it biodegradable? Look for a kind of bond that is fragile chemically. And it actually should stand out to you pretty obviously. There is a bond in the lipid chain that can be broken in the biodegradable FTT5 that's different than the TT3 that's right above it. Okay, so you want to look at these like an organic chemist, and one of the main things that we've already talked about is that when you have a saturated, you have basically a linear, it's got rotatable bonds, but the most stable configuration is it just going in a straight line. But when you have a double bond, when it's unsaturated, you have it kicking out to the side. And think about what happens when these all come together in a membrane or in a lipid particle. They are all packing next to each other. Saturated fatty acids or saturated tails will be able to fit together much better than unsaturated ones. And if you just mix in a few unsaturated ones, it's like their legs kicking out. You know, it's like their legs kicking out and they're kicking the person next to them. And so the lipid next to them sort of gets away from them because they're kicking. And it's not able to be as closely packed. It's not able to be as solid. So unsaturated fats will in general be more liquid than the similar saturated fats. And in fact, this is something they've known about for a long time. So candle makers were some of the first lipid chemists, because what you do with a candle is that you take the fat which your body wants to burn in the mitochondria, and you take the fat out, so you separate it from the solid from the less solid. So that means that you're actually separating out. So in fat, you'll have palmitic, stearic, and oleic acids. Those are the three that you need to know. And palmitic is 16, stearic is 18 carbons. Both of them are saturated. And oleic is 1 unsaturation point. Oleic makes the best candles because palmitic and stearic are too solid. They're hard like soap. So they sort of flake off instead of being moldable. So oleic acid is what you want. And this is how they figured out how to do it. This is from a more than 100-year-old lecture from Michael Faraday, the same Faraday the Faraday constant is named for. And he's turning fatty tallow into candles. Basically, you add base and separate out the glycerin. There's going to be glycerol in all of these. Biologically, the fats are stored with glycerol. So you add base to separate out the glycerin, and we'll talk about that in a second. And then you basically wash everything with sulfuric acid. The fatty acids are going to be an oil on the surface of the sulfuric acid. Everything else is going to be blown apart. And so you wash it again, and you press it between layers of coconut matting. And basically you add pressure by adding water pressure to it. The softer oleic acid will squeeze out through the matting because it's slightly liquid. The hard palmitic and stearic acids that are saturated will actually stay in the middle. And so what you do is you scrape off the soft oleic acid, and then you can mold that as much as you want. The unsaturated oleic acid has a lower melting point because it does this, okay? So when we go through our, we had this infographic about the different kinds of fat, and just remember, we had this before. I'm going to return to it and look at the left side. So if you zoom in on the left side, you can see what butter is. And butter is a lot of fat. You should be used to that idea. The fat is in the form of not free fatty acids for the most part. They're esterified fatty acids, where they're esterified to a glycerol. A glycerol is just 3 carbons. Each of them has an OH. So the OH can react with the acid to make an ester. Again, this is pretty straightforward organic chemistry, but it has 3 OHs, so it can do this 3 times to 3 different fats. And it turns out this is the way the body likes to store fatty acids, not by themselves, but as esters that are 3 packs of that. And so, in fact, you can make a candle out of butter, and I saw this thing on like Instagram or something, and I was thinking, oh, this is exactly what goes on in the mitochondria. Because it might not look like a mitochondria, but you have fats and fatty acids that are here. Basically, you have CH2, CH3 groups that are all together. And what you do is you have them all in a group. With a candle, you light them on fire and you start to ignite them, and you combine them with oxygen in an uncontrolled way. Mitochondria do the exact same things. They're taking these fats, they're combining them with oxygen, they're making CO2 coming out, oxygen goes in, CO2 comes out. Its respiration is the same reaction, whether it takes place biologically or in a candle. But the thing is, we can study, as chemists, we can study candles. We can actually figure out how mitochondria work. So that's why a candle is like a mitochondrion. If you want to look up how to actually do this for yourself, if you want to do it for like a family gathering or something like that, you can make a butter candle. It's basically you're just remolding the butter to be able to, and then you're putting a wick in it, and then you set it on fire. Butter can burn. Butter is a candle, and it's not that different. And then you can take your candle and you take a big bite out of it, and you can really impress people. So there's recipes for this because butter is fat, candles are fat, and the body runs on fat. Fat burns. The other thing is, if butter will oxidize if you add a flame to it, it also will slowly oxidize if you don't add a flame to it, if you just have it in an oxygen-rich environment. And in fact, if you leave butter out on the counter, eventually it will go rancid because it will slowly combine with oxygen. Now, if you just leave it out, and especially if it has more double bonds, the more unsaturated it is, the more easily the oxygen will add on to those particular points, and they'll start to break down the chains. So the fact that this happens shows that oxidation is spontaneous, that you get energy out if you add oxygen to fat. And it also means delta G is, I'm going to wait, this is where I would wait for the class to respond, and everybody would know it, but nobody would want to say it because they don't want to be wrong. But I'll let you say it, so say it to the TV. Okay, whatever it is. The delta G is negative. That's a long time for me to say. Try to participate. You'll learn it better if you are wrong sometimes, actually. Because the way to learn how to be right is to take a risk and be wrong. Okay, so the reaction, and then to correct yourself, I should say. Because it's uncontrolled, in the mitochondria, you don't have to worry about like aldehydes and carboxylic acids forming. But out on the counter, the oxygen can add anywhere it wants to, and sometimes you get partial oxidation, which means you get a carboxylic acid or an aldehyde rather than it going all the way to CO2. You get a COO, you get 3 bonds to oxygen or 2 bonds instead of 4 bonds, which is what CO2 is. And so aldehydes and carboxylic acids are both volatile and they react with your nose, and so it'll start to smell. You're actually smelling this. So butter will go rancid more quickly. Olive oil also will go rancid in about a year. It's got double bonds, and the more unsaturated it is, the more likely it is to go rancid eventually. But solid fats like beef fats won't go rancid. I mentioned how you can buy beef fat in a large container if you're looking for beef fat in the refrigerator section, you're looking in the wrong place. Usually beef fats, lards, things like that are going to be actually out at room temperature because they are less reactive, because they are more solid, they are more saturated. Fewer places for the oxygen to attack. So if you leave your butter out, it'll start to smell. And in fact, rancidity happens to any fat, but actually sometimes it can happen very slowly. For example, there was actually an Antarctic expedition that left behind a fruitcake. And so they recently found it. It was 100 years old, and they actually opened it up. And so they opened it up, and they smelled it. It was wrapped in paper. It looked like this. And it was actually very, very slightly of a rancid butter smell to it. So some of the fats had oxidized. Other than that, it looked and smelled edible. I'm not sure they were brave enough to actually eat it, and I'm not sure I'd recommend that. But that's the way that double bonds get you something that double bonds allow you to oxidize the butter more easily. Mitochondria are very good at oxidizing saturated fats because they have enzymes for it. But again, that's a Biochemistry 2 topic. Okay, so remember that we had some acids that actually had multiple double bonds. Those would be very liquid if they were in triacylglycerides, but they're also used for other purposes. So remember we showed you arachidonic acid, and that's a 20:4 fatty acid. It means it has 20 carbons and 4 double bonds. It's actually necessary for blood vessels within a baby's brain to grow. But it's not important for their membranes. It's important for another reason. And in fact, one of the reasons why babies can't make this unsaturated fat for themselves, it's too complicated for their young body to make. It's a very complicated molecule because it's got 4 different things, and so they have to get it from mother's milk. Their mother makes it for them, and the mother's milk has it in it. And if you're making baby formula to substitute for mother's milk, you need to find a place in nature to get it. And so this is another question I can ask you. Where would you go in the supermarket to try to find some arachidonic acid? One of the things you might think immediately is milk, but that's actually not concentrated enough. You want something that is going to be feeding a baby version of the organism, but will be in a more concentrated way than milk. And they actually purify it from egg yolks. It turns out that chicken babies need this arachidonic acid as well, and so the chickens make it, And because life has these things in common, we can take it from the chicken baby format, and it's exactly the same molecules, the 20:4. The reason why you need 20:4 is because you make those double bonds react. Instead of reacting them randomly with oxygen, like in butter, you have enzymes that specifically react them. In some cases, they still oxidize them. So you can see you have arachidonic acid up here. They fold it in half, and that brings the double bonds next to each other. And you know from organic chemistry, you can do a lot of stuff with double bonds like that. So these actually, you can take arachidonic acid, you can make this prostaglandin, and if you've heard about biology, you might have heard of prostaglandins as signaling molecules. You can also make thromboxane, another signaling molecule for a different signal, or you can make leukotriene. All of these send different signals, and in fact, right here, these prostaglandins are what cause fever and pain. They signal the immune system. And you might want to block that. If you don't want a fever, you might want to block the fever signaling molecule. So the enzyme that turns arachidonic acid into prostaglandin can be blocked by aspirin. And this is one of the ways that aspirin or NSAIDs, other drugs that will block this, will block fevers. It's very cool and very important stuff. So all of them that I've been showing you have been natural fats, and so remember that for the unsaturation points, the natural configuration is going to be the cis arrangement of the bond. But if you have the trans arrangement, so they start out that way, and by the way, there is a little bit of the trans arrangement, but it's in the middle of an enzyme. Again, you'll talk about this in Biochem 2. So it's not floating around in the system. It is present technically, but it's not floating around in nature. Okay? When you don't have nature, when you have a factory, people figured out pretty early on, double bonds cause it to be liquid versus solid. So what if we have an oil, a cheap oil liquid, and we want to turn it into a cheap substitute for butter? We just want to make it solid, right? So what they did is they actually just took oil, which has double bonds, and they bubbled hydrogen through it. And the hydrogen sort of combines, and it saturates the double bonds, and then you end up with solid margarine. It's exactly how margarine is made. The only problem is you aren't controlling this reaction, and so sometimes you get the hydrogens going on and off, and some of the fats get flipped around by this. So they get flipped around by, there's still a few double bonds left, and some of those double bonds get flipped around to the trans configuration, and the body doesn't know what to do with that orientation. That structure is not a natural structure, so the body hasn't dealt with it. They're also more stable than cis. And so because you're doing this in an uncontrolled way, you will get the unnatural, more stable form of the bond. And they're particularly bad for your arteries. Actually, later we'll talk about a little why, but we have to get to the membranes chapter before we talk about why. So that means that you have this infographic, and just remember that I like this infographic because it does show you the polyunsaturated fats are in the cis configuration, and when you flip it around, when you're doing it in the factory and you flip it around, you get the trans configuration. And you see how that has a different structure. That's no longer going to be the kicking out structure that you had before. That works well for making it more solid. It doesn't work well for interacting with other parts of the body. All right, so that is our first mini lecture. The next one, we're going to talk about chocolate instead of candles.